Hey, how you doing? Today's topic is going to be transport across cell membrane from general physiology. Let's get started. Now, as far as your theories and practical vivas are concerned, this topic is relatively important, so pay close attention. Today's topic will be discussed under two broad subheadings, the introduction and later the types. Towards the end, we'll summarize the entire lecture for you and then we'll tell you how to write an answer in your exam if this question comes. Okay, so introducing this topic, before we actually dive into the transport mechanisms and how they function and what role do they play in our body, it's important to understand the basic structure of cell membrane. Because transport across cell membrane requires the knowledge and understanding of the basic structure of cell membrane. So according to the fluid mosaic model, the cell membrane is chemically composed of three components: the carbohydrates, the lipids and the proteins. The carbohydrates are usually present along with the proteins as glycoproteins, carbohydrates and protein complex. The lipids are present as bilayer and proteins are present in between the lipid bilayer or on either side, either inside or on the outside. So this divides the proteins into two types. The proteins which are present across the membrane or the transmembrane proteins are called integral proteins, and the proteins which are present either on the inside or on the outside are known as peripheral proteins. But the most important component out of these three which play a role in transport are the proteins. So the main functions of the proteins are cell adhesion, which help in adhesion of the neighboring cells, not really important for our topic right now. Transport. These proteins can act as channels, carriers or pumps. Now these three terms are similar but they are very different. They function differently and they play a role in different transport mechanisms. So protein can either act as a channel or a carrier or a pump. Protein can be any one of these, not two or all of these, right? So moving on to the next function. These functions will be described as the lecture proceeds as you dive into the various mechanisms of transport. The next function is they act as receptors. Now we know that hormones in the body act via receptors which are present on the cell membrane or within the cell. That's a topic for a later time. But important concept here is that the receptors which are present on the cell membrane are the proteins which are there. So these proteins may act as receptors as well. Some of these proteins have enzyme activity as well. So these are the basic functions of the proteins. Now we'll talk about the various types of transport mechanisms and the role of different proteins as channels, carriers and pumps. So now that we've seen the basic structure of a cell membrane, let's see the various types of transports across cell membrane. Transport across cell membrane is basically divided into three types: passive, active and vesicular. The word passive in itself literally means two things. The first thing here is that no energy is required. In passive transport, no energy is required. That's important. And second, the movement of particles or molecules is from an area of its higher concentration to an area of its lower concentration. These two features or these two characteristic features define the passive transport mechanisms. Passive transport mechanism is further of two types: simple diffusion and facilitated diffusion. So it's really obvious that both these, simple and facilitated, require no energy and movement of molecules takes place from a high to low concentration. Moving on to active transport. The word active literally means two things again. Energy use is required, and secondly, the movement of particles is from low to high concentration, that is across the electrochemical gradient or uphill. This is along the electrochemical gradient or downhill. Active, energy utilized, and this is uphill or against the electrochemical gradient. Active transport is further of two types, primary and secondary. Primary active transport basically uses ATP as energy source, and secondary active transport basically uses the sodium gradient as energy source. So these are the basic differences in the two types of active transport. Both these transports will use energy, and both these mechanisms will cause the movement from low to high concentration, but the utilization or the source of energy is different. In primary active transport it is ATP, and in secondary active transport it is the sodium gradient. Now gradient basically refers to the difference in concentration. For example, if this is a semi-permeable membrane, this is side 1, this is side 2. If sodium is in high concentration here, it will definitely move from an area of high to low concentration. So this difference in the concentration is what we call as the sodium gradient. So sodium gradient is secondary active transport. Vesicular transport is of two types, endocytosis and exocytosis. Endocytosis means movement of particles within the cell from outside to inside the cell. This is endocytosis. From outside to inside. Endo means inside. Cytos means cell. So movement of particles is inside the cell. Exocytosis means definitely outside the cell. Exo means outwards. So movement of particles is from within the cell towards the outer side of the cell, right? Endocytosis is further divided into phagocytosis and pinocytosis. Phagocytosis, as you might have heard the term that the WBCs and the macrophages phagocytose the bacteria. So phagocytosis is ingestion or movement of larger particles like bacteria or solid particles within the cell. The cell can be anything like WBC, neutrophil or macrophage. So when a macrophage, if this is the antigen and if this is the macrophage and it is ingesting the antigen, this is phagocytosis. Okay, so solid particles moving inside the cell is phagocytosis. Pinocytosis is also called cell drinking. It means the cell ingests certain substances which are in the solvent form, right? So solvent form moving into the cell is pinocytosis. So phagocytosis and pinocytosis, they both are endocytosis because substances are moving inside the cell. Exocytosis is outward movement of the cell, right? So now we'll discuss in detail each of these mechanisms separately, starting from simple diffusion, facilitated, moving on to primary and secondary active transports. Then we'll talk about endocytosis, phago and pinocytosis, and we'll end by talking about exocytosis.